structures. We are discussing diplotin. Diplotin, in this one, what initially what happens? So homologous chromosome starts separating. Homologous chromosomes start separating. Homologous chromosomes start separating, except where crossing over occurs. These are all separated like this way. Except this. Except this means what? Where crossing over happens. Except these, remaining all are separated. These are all separated. These chromatid are separated, except where this is the place where crossing over happens. So this letter, due to crossing over, these teams seems to be a X letter. These X-shaped structures are called chiasmata. So it resembles X-shaped letter. So these X-shaped structures are called chiasmata. Chiasmata. See here given. Okay, chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. That means what? In oocytes of some vertebrates, those crossing over, where the crossing over occurs, those crossing over where occurs, those chromosomes are remains attached in X-shaped structures only for months to years. In some species, after 1 or 2 days, that crossing over where happened, those chromosomes are separated. But in oocytes nothing but ova of some vertebrates. Diplotin stage can last for months to years. Okay, that's it. See here, clearly if you see, get to know that, see chiasmata. See chiasmata here. See, these are centromere. It is one chromosome. This is one chromosome. Where crossing over occurs, except that place, remaining are separated. See, this chromatid separated. So chromosomes with 2 chromatids. See, chromosomes with 2 chromatids. One chromatid, it is one chromatid. Okay, see, 2 arms of one chromatid. See, 2 arms. One, it is one arm, it is one arm. 2 arms of chromatid. These are chromatids. Okay, chromatids. These are all chromatids. Okay, so here except where crossing over occurs, remaining are separated. This is main theme of diplotin. Next topic. Diakinesis. Next one is diakinesis. It is the final stage of meiotic prophase 1. See here we will discuss that. So prophase 1, metaphase 1, prophase, metaphase, anaphase 1, telophase 1. So this prophase 1 again divided into 5 sub: leptotin, zygotin. Third one is pakitin, diplotin. Last and final one is diakinesis. Okay, it is the final stage of meiotic prophase 1. So the chromosomes are fully condensed. In this one, what happens? All the chromosomes are fully condensed. See here, chromosomes are condensed. Condensed means what? Like this way, chromosomes in last stage condensed. Condensed means thickened. The chromosomes are fully condensed, and the meiotic spindle is assembled to prepare the homologous chromosomes. See, meiotic spindle fibers starts growing. Those are all comes together to prepare the homologous chromosomes for separation. So with the help of spindle fibers only, homologous chromosomes are starts separation. So for that, what happened? Homologous chromosomes start moving towards equators. Okay, so initially, so chromosomes are like this way formed. Gradually, the chromosomes homologous chromosome starts pairing. like this way, like this way. Homologous chromosome starts pairing. These are called homologous chromosome starts pairing and slowly move towards opposite poles. So some chromosomes are moved towards this side, some chromosomes are moved toward opposite side. Okay, that's all. So homologous chromosome starts moving towards equator. Sorry, ma, here little correction. Equator means what? Homologous chromosome starts moving equator. Equator means what? Like this way. Chromosome comes to center. Equator means what? Middle, like this way. Homologous chromosome starts moving towards the center. Nuclear membrane disappears. See, around the chromosomes, whatever may be the nuclear membrane is there, that nuclear membrane is disappears. Centrioles migrates to poles. Centrioles from center to they are moves to opposite poles. These are called centrioles. Centriole moves to opposite poles. Spindle fibers begins to form. From them, spindle fiber slowly begins to form. Spindle fiber slowly begins to form. By the end of diakinesis, the nucleolus disappears. Inside the nucleus, the nucleolus will be there. That nucleus is disappears. And the nuclear membrane also breaks down. Nuclear membrane, this is the nuclear membrane. The nuclear membrane is completely breaks down. These are all the changes takes place. See here. Those are all here shown. See, slowly what happens, see here. Homologous chromosome starts pairing. Pairing occurs. Homologous chromosome starts pairing. Next, those homologous chromosomes moves towards the center. Okay, this is we are discussing diakinesis. So actually that diagram also here how to show, but they did not give. Okay, so these are all comes to I have drawn. They are moved to center. So these are all centrioles moves slowly towards the. It is already settled at the opposite pole. It is also comes and settle exactly towards this. Okay, these are all the changes takes place in diakinesis. Next, prophase 1 with this successfully finished. Next one, what we have to discuss, metaphase 1. Next, after prophase 1, like mitosis only. First one, what? Prophase. Prophase 1. It is again divided into 5 subphases. Leptotene, zygotene, pachytene, diplotin, and diakinesis. Now, metaphase 1. Okay, see, bivalent chromosomes are aligned at equatorial plate of the spindle. See, bivalent means what? Like this way, homologous chromosomes. Homologous chromosomes. These are called homologous chromosomes. Homologous chromosomes are aligned, settled at the equatorial plate of the spindle. See, equatorial plate means what? Middle, exactly. This is the cell, middle, they settle. Okay, bivalent attached at the centromere to the spindle fibers. So these are bivalents. These two, these two, these two, these two. So bivalents are attached to the centromere. Attached at the centromere to the spindles. See, bivalent chromosomes are attached to the centromere. From the centriole, spindle fibers are formed. Those spindle fibers are attached here. Those spindle fibers are attached here. Here, just they given one pair of chromosomes only. Like this way, many pairs of chromosomes will be there. Many pairs, if they give means the diagram we cannot draw. That's why they will represent one chromosome. Bivalents are attached to the centromeres. See, these bivalents are attached to the centromeres through spindles. So next, the microtubules from the opposite poles of the spindle attached to the pair of homologous chromosomes. Here, one point you have to remember. What is that? It is the cell membrane. Here, centriole commonly will be there. Here, centriole commonly will be there. So, these are spindle fibers. Centriole forms spindle fibers. Along with the spindle fibers, centrioles also arises a microtubules. See here the microtubules. So some microtubules also arise. See here those microtubules. These are called microtubules. From the centrioles, microtubules are also arising. See the microtubules from the opposite poles of the spindle fibers attached to the a pair of homologous chromosomes. See, microtubules means what? So from this centriole, microtubules are formed. From this centriole, microtubules are formed. These microtubules are attached to the pair of homologous chromosomes. See, these are microtubules. See, these are microtubules. Microtubules are attached to the homologous chromosomes. Some microtubules are without attaching, they will be moved towards opposite poles. Some microtubules, some spindle fibers. See, 1, 2, 3. These spindle fibers did not attach to any type of chromosome. Okay, so like this way, some microtubules also moves towards opposite poles. Like, see, spindle fibers from here started, from here started, both are attached. From here started, here started, both are attached. These are called spindle fibers. Okay, so that's remaining. If you observe labels, these are all homologous pair. Homologous pair. To this homologous pair, some microtubules are attached. Spindle fibers are attached at the centromere region. This is called metaphase explanation. Metaphase explanation. Okay, that's all. Next. Next, anaphase. Next topic is anaphase. Okay, ma. Anaphase. See, are you getting my voice now clearly, ma? Dear students, telophase. Here, what happened? Cytokinesis division starts. Cytokinesis 1 start. So here, no need to mention. We have to understand. Here, as a result of cytokinesis, only these two daughter cells are formed. Actually, specifically, this division, no need to mention. Next, you see, before discussing about meiosis II, here a brief review we will see here. See here, brief review in the sense what? See here now onwards. Metaphase 1, anaphase 1 I finished. Telophase might be here onwards. Anaphase. So anaphase 1, what happened? Spindle fibers pull the whole chromosomes. Spindle fibers, see, these spindle fibers pull the chromosomes. These spindle fibers pull the chromosomes. These chromosomes are moved to opposite poles. So gradually the length of the spindle fibers is decreased. At the end of the anaphase 1, so these are all the chromosomes are moved to opposite poles in this direction. So if you will consider this is north pole, if you will consider this is south pole, so completely chromosome movement is finished to their north pole and south pole regions. Okay, that's all. Next here, telophase 1. So in this one, what happened? Each pole possesses chromosomes composed of two chromatids. See, this pole, in this region, chromatids are present. In this region, chromatids are present. So when telophase 1 begins, spindle fibers are completely disappears. Next here, these are chromatids. These chromatids are uncoiled. See, like poles only, those are present, not coiled. So forming chromatin network. After some time, these are all the chromatids are turned to be chromatin-like structures, like these way structures. Here, opposite pole chromatin-like structures. Nuclear membrane, nucleolus are reappear. See, this is the nuclear membrane, nucleolus are reappeared. Okay, that's next here. As a result of that, what happens here? Cytokinesis 1. Cytokinesis 1. Okay, so all the intracellular organelles are formed after the finishing of telophase 1. All the intracellular organelles are reappeared at the end of the telophase 1. At that moment here, so this is cytoplasm. This is called cytoplasm. So in the cytoplasm, so this is cytoplasmic membrane. This is the membrane. The cell membrane slowly forms a thin wall. A thin wall. This wall gradually develops and matured. After maturation of this wall, so exactly middle of the cell, this wall is break or cut off and divides these two cells. As a result of meiosis 1, so leptotin, first we have seen leptotin, it is a single cell. So now as a result of meiosis 1, single cell is divided into two cells. This is one daughter cell, this is two daughter cells. So these two daughter cells are formed as a result of cytokinesis 1. Okay, so after these divisions, interkinesis, these two cells undergoes to interkinesis 2, and again for further meiosis 2 divisions continued. So for further meiosis II divisions are started with interphase 2 divisions. Okay, so these two cells undergoes resting period. During the resting period, inside the cell material is ready to perform meiosis II divisions. Okay, that's all. So here it is initiated immediately after cytokinesis 1. So meiosis II divisions immediately starts after cytokinesis 1, usually before the chromosomes have fully elongated. Okay, before the chromosomes fully elongated. So chromosomes after some time they are elongated and starts homologous pairing. So before that only meiosis II division started. So meiosis II division started. Before starting meiosis II, chromosomes are in like this way, chromatin like structures. Chromatin like structures means thread like structures. These threads gradually turn to be rod like structures. Okay, that's all. So before the chromosomes have fully elongated. So chromosomes fully elongated means what? Like this way, chromosomes elongation starts. Okay, that's all. In contrast to meiosis 1, meiosis 2 resembles a normal mitosis. Here one point you have to remember. So meiosis 2 divisions are similar to mitosis. Okay, so whatever the divisions in happen in mitosis, those are all repeated under meiosis 2. Okay, so meiosis 2 means it is not any separate. Okay, so as a result of cytokinesis 1, what happened here? Two daughter cells are formed. Two daughter cells are formed. Those two daughter cells are these. So its division starts like this way. Okay, so mitosis divisions are similar to meiosis 2 divisions. In mitosis, whatever maybe the divisions happen, same like that meiosis 2 happens. And finally what occurs here, see, so here too, as a result of meiosis 1, two cells are formed. Those cells undergoes prophase 2, metaphase 2, anaphase 2, telophase 2, and finally cytokinesis. Here see telophase, here telophase 2. So this is the anaphase 2. Anaphase 2 becomes 4 cells. So here meiosis 2, when the anaphase starts here like this with 2 cells, it is 1 daughter cells, 2 daughter cells, 3 daughter cells, 4 daughter cells. In between these two, a thick layer is formed. In between these two, thick layer is formed. So that thick layer is gradually developed, matured, and cut off. Then it becomes 4 cells. See here, it becomes 4 cells. See here, 1, see here, it is 1, 2, 3, 4. Okay, like cytokinesis 1 how happened, cytokinesis 2 also happened. As a result of meiosis 2, totally 4 daughter cells are formed. 1, 2, 3, 4. With these meiosis 2 divisions are finished.